Today we're going to be talking about the structure of DNA, and I mean that's our learning objective, right? So you're going to talk about the chemical components of DNA, what clues led to the discovery of the structure of DNA, and then just a little bit about the double helix molecule. This should look familiar to you. We've already discussed it a little bit, but this is called nucleotide. DNA is made up of nucleotides joined together in long chains of covalent bonds and then strung across from each other. If we're looking at the nucleotide, it's got a nitrogen base, a 5-carbon sugar called ribose, and then the phosphate group. Again, this should be familiar to you. The nitrogen base can be one of 4 different nitrogen bases. It can be adenine, guanine, cytosine, and thymine. And you can actually see here, the picture does a good job showing you how the nucleotides go together, right? So they're strung on top of each other, where you have phosphate, sugar, nitrogen-containing base, then the next one, phosphate, sugar, nitrogen-containing base, and so on. Talking just quickly about the nitrogen bases. There are 4 kinds of nitrogen bases, and they're split up into 2 distinct groups. You have the purines and the pyrimidines. The purines have 2 rings, like you see here. Adenine and guanine are purines, and thymine and cytosine are pyrimidines, and they just are made of 1 ring. And the way the DNA or the nitrogen bases bond together, so that they're equal in size, there's always 1 purine and 1 pyrimidine put together, and it's always the same distinct pairs that go together. So A goes with T, C goes with G, or adenine goes with thymine, cytosine goes with guanine. Pointing out, some other parts here. Again, we see 1 nucleotide here. We also see these little bonds between the ribose and the phosphate. That's called a covalent bond. We've already learned about covalent bonds earlier in the year. And then something else I want to point out, you'll notice that there's a consistently repetitive structure on the outside with the phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. It goes down the side of the DNA molecule just like that. That is called the backbone of DNA. Sometimes they also call it the sugar phosphate backbone. And then the middle is the center of the DNA molecule. And this is how they are strung together. So we have single nucleotide here, another single nucleotide here across from it. We have this alternating phosphate, sugar, phosphate, sugar, phosphate, sugar on the outside. Same thing happens here, but if you look, they're flipped upside down. So one goes in this direction here, and this other side looks like it's been flipped upside down. I'll explain this more when we get to replication, but this has to do with where the phosphate is bonded to the ribose sugar here. And so one direction, it's said to go from the 5' direction to the 3' direction, like we see here. The other side, if we're going from up to down, actually goes in 3' to 5'. So again, it's upside down and flipped. We'll talk a little bit more about this in our next little section, but there is a reason for that. And so they are called anti-parallel strands. The other thing to point out again, nitrogen pairs, again, always 1 purine with 1 pyrimidine. Adenine always goes with thymine, or A with T, and cytosine go with guanine, or C with G. I remember it. A and T are both like linear in structure, right? They're both pointy, and C and G both basically have the same shape. So C and G go together because they look alike, and A and T go together because they look alike. And then what bonds these 2 strands in the middle is called a hydrogen bond. Last little bit here in our notes is about the discovery of the DNA molecule. So this is called, her name is Rosalind Franklin. Rosalind Franklin is one of the scientists who helped to discover the shape and the structure of DNA. She did this procedure called X-ray crystallography. And X-ray crystallography was basically a way of turning large molecules into crystals and then using X-rays to shoot through the crystals to try to visualize the 3D shape of those molecules. It was a pretty revolutionary science when she was doing it. Um, and this is her most famous picture. It's called Photo 51. And this basically displays a DNA molecule if you were looking down on it, and it shows an X figuration. And that's the hallmark trait of a double helix molecule. And DNA is exactly that. It's a double helix. It looks like you had a ladder and twisted it. Sometimes it's also called a twisted ladder shape. I bring Rosalind Franklin up because oftentimes she's not credited as much as the next two people we'll talk about, Watson and Crick. But without her work and her picture, Photo 51 here, that was basically taken without her knowledge, Watson and Crick would not have been able to put everything together here. So Watson and Crick are the scientists who end up getting most of the fame for their work. They had been working on the structure of DNA for a while, and they were down to a few different shapes. And they kind of stole Rosalind Franklin's work without her knowledge, and used her work along with the work they'd been doing to put together that the structure of DNA was a double helix molecule, right, where you have two strands of nucleotide sequence bound around each other. So that's what we have. That's the DNA molecule. That's the in-depth structure. It's made up of nucleotides. You have two strands of nucleotides. One runs this way, the other one runs this way in anti-parallel strands. Nitrogen bases in the middle basically act as the rungs of the ladder. And then the phosphate sugar on the outside as the backbone. Then we have the hydrogen bonds in the middle that connect the nitrogen bases. Okay.